Hello, my name is Dr. Diwan S. Raja. Today I will discuss about the histology of the skeletal muscle. What are the identifying points? Skeletal muscle cells are long, multinucleated fibers. They are cylindrical in shape and nucleus is located just under the cell membrane. So it is subcircular nucleus. We have multiple nucleus in each cell, so these cells are multinucleated cell. There is cross striation due to presence of actin myosin. The sarcolemma or cell membrane of the cell is surrounded by endomysium, and the skeletal muscle cells are also called skeletal muscle fibers. Okay. So each muscle fiber is surrounded by the endomysium. So here the muscle fiber, this is surrounded by endomysium here. The group of fiber are called fascicle. They are surrounded by the perimysium. This is perimysium. The muscle fascicles are together surrounded by the epimysium. This is the epimysium. These are connective tissue. They carry the blood vessels, nerve, and lymphatics to the muscle fibers. Okay, we have some terminology for the muscles. Cell membrane is called sarcolemma. Smooth endoplasmic reticulum is called sarcoplasmic reticulum. Cytoplasm is called sarcoplasm. Muscle fibers are often used as muscle cell. Muscle cell is used as muscle fiber. Sometimes mitochondria is called sarcosome. We have another type of cell that is called satellite cell or undifferentiated reserved progenitors, myoblast, located on the external surface of the muscle. We know that our muscles, the skeletal muscle, is multinucleated because embryologically it develops from fusion of multiple myoblasts. These myoblasts are derived from the mesenchyme, that is the embryonic form of connective tissue. Some of the myoblasts are they remain undifferentiated, act as a reserved progenitor myoblast on the external surface of the muscle. If the muscle is damaged or injured, or in case of vigorous physical exercise, some of the satellite cell will be the differentiated skeletal muscle cell. Okay. Skeletal muscles at higher magnification, if you look at the skeletal muscle, this is the muscle. Okay. This is muscles of the thigh. Okay. Suppose this is sartorius muscle. Here, with the muscle, this is surrounded by the epimysium, bundle of muscle fibers called the fascicles surrounded by the perimysium, and individual muscle fibers surrounded by the endomysium. Muscle contains the myofilament, contains the myofibrils. Myofibrils contains the myofilaments. So we'll look at that. Each muscle fiber is filled with myofibrils composed of thousands of thick myosin filament. This is the thick myosin filament and thin actin filament. They are highly organized into contractile units called sarcomere. Sarcomere extend from the middle of the thin filament area, the Z line, one Z line to another Z line. Okay, the Z line is located in the center of the thin filament. So this is the sarcomere from one Z to another Z, and this sarcomere is the structural and functional unit of the muscle. Within the sarcomere, thick and thin filaments interdigitate. Globular myosin head projects from the thick filament towards the F-actin filament, which is associated with tropomyosin and troponin. Okay, so we have thick filament, thin filament. Thick filament is the myosin, the thin filament are the actin, the troponin, and tropomyosin. So there is sliding taking place between the thick filament and thin filament. So what happened? Myosin head projects from the thick filament towards the F-actin filament, which are associated with tropomyosin and troponin. F-actin filament, where the myosin head is bended during muscle contraction. Okay, so organization of the skeletal muscle is here. We got that. Here we are looking at the AI junction between the actin myosin. We have the triad. This triad composed of T-tubule, that is the invagination of the cell membrane, that is the sarcolemma, and it is in contact with that of the terminal cistern of the sarcoplasmic reticulum. There is a smooth endoplasmic reticulum. So there is a source of calcium. Calcium is diffused out. Impulse goes through that, and there will be muscle contraction. Okay. So we have to know what is triad. Triad has three components. One on this side, the cistern, the sarcoplasmic reticulum cistern. On this side, sarcoplasmic reticulum cistern. In between, we have the T-tube. We have thick and thick filament. Thick and thin filament are present in the myofibrils, sarcomere, from one Z line to another Z line. Between the myofibrils, we have the organelles like mitochondria. Okay, we have the nucleus that is present just underneath the cell membrane. We have multiple nucleus. We discussed that the skeletal muscle cells are formed by the fusion of multiple myoblasts during the embryonic life, so that we have multiple nucleus. One is here, another nucleus is here, and this is the triad that is composed of one of the T-tubule and two sides we have the cistern of sarcoplasmic reticulum. T-tubule is an invagination of the sarcolemma. Okay. Here, composition of thick and thin filament. Thin filament has multiple components like that of the actin, as again we have the G-actin, F-actin, and we have the tropomyosin and the troponin. Troponin has calcium binding sites. Once it is binded to the calcium, then the F-actin area will be exposed from the tropomyosin so that the myosin head can bend over the F-actin, so there will be muscle contraction. So for muscle contraction, we need calcium binding and exposure of the F-actin part from the tropomyosin. So troponin, tropomyosin, along with calcium is essential for the movement of the head. This is going to be bended head of the thick filament, the myosin, bended over the F-actin. Okay, so here if you look at that muscle contraction, this is the muscle relaxation here. Here is the Z disc. From one Z to another Z is the sarcomere, the unit of the functional unit of the and structural unit of the muscle. And we have the thick filament. In the middle of the thick filament with the H band. In the middle we have the M line. So what happened during contraction? This goes inside this. This goes inside that. So we are looking that here the thin filament is moving towards the M line, on both sides so that the sarcomere length will be decreased and muscle will be contracted. There is the idea. So we have the M line here, Z disc. From one Z to another Z is the sarcomere. This is the biceps brachii muscle is relaxed. Biceps brachii muscle, skeletal muscle, that is contracted. So where we'll get the skeletal muscle? It is related to the skeleton like biceps brachii, triceps brachii, deltoid. All of them are skeletal muscle. Plus the tongue is a collection of skeletal muscle. The upper one-third of the esophagus is composed of skeletal muscle. The diaphragm separating the thorax from the abdomen is composed of skeletal muscle. It is nerve supply somatic, voluntary. Phrenic nerve supplies the diaphragm. Okay, suppose biceps brachii supplied by the musculocutaneous nerve that is coming out of the brachial plexus. Okay, highlights identifying point: multinucleated, cross striation, cylindrical muscle cell, and that is the skeletal muscle. These are very long. Some of the muscle cell may be several centimeters. Sartorius muscle, very long muscle. We have the muscle fiber, very long. Multinucleated muscle cell, sarcolemma. Okay, so sarcolemma is the cell membrane. Myofibrils, that is present inside the muscle cell, and the endomysium. Endomysium is the connective tissue covering around the myofibril, around one muscle fiber, endomysium. So around one fascicle, we have the perimysium. Around the entire muscle, we have the epimysium. So epimysium, perimysium, endomysium. Sarcomere extend from Z line to Z line. Z disc is present in the middle of the thin filaments here. Okay, this is the thick filament. Okay, so from Z line to Z line, we have the sarcomere, and this go inside that, and the sarcomere length will be decreased. So during muscular contraction, this is the muscle, and we have the myofibrils here, and muscle fiber is surrounded by the endomysium, and the muscle fascicle surrounded by the perimysium. Entire muscle is surrounded by the epimysium. Okay, M line is present here in the center of the thick filament, and this is called H band area in the center of the M line. T tubule is the invagination of the sarcolemma, and triad composed of the T tubule on each side will get the sarcoplasmic reticulum cistern. And our skeletal muscle has limited regenerative capacity, but we have satellite cell due to vigorous exercise in the young age, and also during injury during the young age, some of the satellite cell may be converted into the skeletal muscle cell and they contribute in the skeletal muscle cell. Okay, so these are my references, and that's all about the histology of the skeletal muscle. If you like my video, please support my channel. Please share the information with your friends. Any question, please feel free to ask me, and have a nice day. Please subscribe me. Bye now.